the John Innes back in the 80s, I was really studying plant viruses, and really from an academic point of view. And that actually came from an interest I had all the way back when I was an undergraduate in Cambridge in biochemistry. And this is where it all began. And yes, folks, that's me. I haven't changed a bit. We did everything in black and white in those days. That was in 1976. And I was still an undergraduate, and this is where I got my love of plants. This is a tobacco plant, as you probably recognise, during my undergraduate project. And I got fascinated, and I was really interested in the sort of intricate details of molecular mechanisms. And in those days, it's all pre-PCR and genome sequencing. Really, the only way you could get a handle on some of the details was to actually work on a virus and use that almost as a probe for functions. And so I did my PhD on the self-assembly of tobacco mosaic virus, which is the grand old man, woman, whatever you want to have it, the grand something of virology. And in fact, it was the entity for which the name virus was originally coined. So I started my PhD, and this was my first ever paper in 1978. And it's not exactly a riveting title, I don't think. It was good at the time. And you might think, well, I'm going to talk about, applications and things. So I must have maybe had that in mind when I was doing my PhD. And this is what it was on, how this particle assembles. And actually, to bring it up to date, this is a 3D model of TMV, which I'll hand to my lovely assistant, who will pass amongst you, or pass the model amongst you, a 3D model of that virus particle. And basically, we're looking at how that interacted specifically with the RNA, the genetic material. And was there any commercial potential that we foresaw? Were we visionaries at the time? And the answer is, not that I could see. And people used to sort of, rather to take the mickey out of me for this, particularly as I did these studies on this plant virus at a medical research council facility. And people were saying we ought to be trying to kill tobacco, not sort of cure it. For health reasons. Anyway, some of the techniques, would just seem completely from a different era, the kind of way we did that. The genome is 6 kilobases, and we were nowhere near even being able to determine the sequence of that. We had little short segments, that's all we had of the sequence back in the 70s. And fast forward, I'm going to go straight through the 80s, vast amount, just complete change with all this rapid DNA sequencing methods, synthetic oligos, PCR, of course, came in early 80s. And so got a huge amount of additional information about the genome structure, a lot of work on crystallography of viruses. You get actually 3D images of whole virus particles. Began in the very late 70s, but really took off in the 80s. Means of genetically manipulating reverse genetic systems through infectious clones, so you could actually make mutants of your desire. Even in the 70s, if you want to make mutants of a plant virus, you just treated the RNA with nitrous acid, and you got what you were given, basically. It may or may not have had interesting properties. But now you could deliberately manipulate particles. And so this led to the thought, well, maybe you can actually try and exploit these properties of the virus, small genome, very defined particles they make. And I'm really only going to talk in detail about one particular virus, and it's my second love after TMV, and that's this virus called Cowpea mosaic virus. And what we wanted to do initially was to try and use some aspects of that virus to make pharmaceuticals in plants. And this is there you see your pharmaceutical vaccine dripping out of the end of a leaf in a rather an illustrative image of it rather than a scientific one. And so this is the virus, and the details are not that important, but what I will again pass around is a 3D model of the particle structure. This is Cowpea mosaic virus, completely different shape. The other main class of shapes, which is isometric or spherical, and basically it infects both cowpeas and gives a mosaic, hence the name. Black-eyed peas, those are black-eyed beans, also affects Nicotiana benthamiana, and this is what it looks like under standard electron microscopy. And it has 2 RNA components, and again, don't worry too much about the details of all this, but it's worth actually mentioning it has an RNA which encodes all the features necessary for the replication of the viral genome, which is RNA, not DNA. And it has a second RNA, which encodes structural proteins, particularly the large and the small coat protein. So structural proteins are hived off onto this separate RNA, all the replication enzymatic functions on this RNA, and this can't do anything, this can't replicate on its own, it needs these functions. And you can separate these into different components depending on empty particles, middle, and bottom. So there's the middle have RNA2 and the bottom of RNA1, they're heavier, a larger RNA. And here's the detailed atomic structure, that's the model going round, and it's solved in detail, you can see it's mainly beta sheet, and basically there's 60 copies of what's known as the asymmetric unit clipped together to form those isometric particles. And this is what the particles look like. I'm not sure this really tells you an awful lot that you can't see from the model, but this is a recent cryo-electron microscopy reconstruction of the particle. And I'll just point out a couple of things here. You can get really nice atomic resolution structure. You can see amino acid change, the beta strands. But one thing I want to show you, this gold in the center, that's the genome. You can't see that much detail because it doesn't have the symmetry of the multiple copies of the coat protein. But right in the center, you can actually visualize that by cryo-electron microscopy. So that's what the particles look like, and that's what that model being passed around is actually based on, the coordinates from there. So years ago, this is into the 90s now, just had this idea that, well, we know the structure of the particle in detail, we have means of manipulating it. Could we actually do something with this knowledge, something useful? And the idea is you could have this, use the particle as a sort of scaffold to put different antigenic sites, different immunological sites on the surface, and select those from an animal virus. And then if you supplied that to an animal, and potentially even a human animal, you might be able to get an immune response against the bits you've inserted. And so this is, I deliberately keep this old slide. I mean, it's the old technology with literally a slide, which I've now copied. You can see the work in the 90s, 25 years ago, a lot of this. And basically, this is very, I mean, you don't use this technology anymore, but you insert a bit of a sequence into the DNA, infect plants, and you get your modified particles out. And this is the kind of thing you get. And this is a crystallographic image. So this isn't just a model. This is actually a structure where you can see that in the green and the blue is the actual Cowpea mosaic virus scaffold, the thing I've just passed around. But in red on it are part of the common cold virus grafted on, and so 60 to the small coat protein. So there's 60 copies each of that small common cold virus sequence on the outer surface. And if you do that you can make all sorts of things like this, all sorts of foreign sequences. And the one we particularly worked on was a thing called mink enteritis virus, which is a disease of farmed animals. And this is a mink. In fact, it's a stuffed mink, which I borrowed because it's easier to photograph them when they don't move, and they don't bite you either. I don't think they live in woodland particularly, but that's a rather sort of fancy image. You won't, I'm sure, really be able to see this. The presentation has been copied. But basically, if you put this chimera, this Cowpea mosaic virus scaffold with this small bit of mink enteritis virus, inject it into mink, and then you challenge them with real mink enteritis virus, they don't get sick. So you made a plant-based vaccine against an animal disease. And that was very exciting at the time, again, over 20 years ago. And the issue was that using a whole virus, viral infection, there's limits to the amount you can express on the surface. And this would work fine for small sequences and diseases which you only need a very small section of amino acids to elicit protective immunity. But that's not always the case. So we really wanted to have a look at what else we could actually do. Could we improve the expression system we use rather than just using infection? And after many trials and tribulations, shall I say, it hit on this system. And this uses Cowpea mosaic virus again, and it's a deleted version of RNA2. And if you remember, that's the RNA which encodes the structural proteins and is dependent on the other RNA for its replication. So on its own, it won't be able to multiply. But what you can do is take the genome, and you can actually take most of the coding region out. And what you're left with is a deleted version, which is basically looks like a messenger RNA, an effective messenger RNA. And you can put a foreign sequence in, and of course you always try, and you may well have done experiments on this, on green fluorescent protein, because it's great, it's visual, and it always works. So GFP always works, which can be misleading. And we use a technique called agroinfiltration, which I'll explain a little bit more about in a minute, just to take the bacteria into the Benthamiana leaf. This is a variety of tobacco, sometimes known as Australian dwarf tobacco. Interesting, just a little aside, it was a variety collected on the third voyage of HMS Beagle, which of course is most famous for its second voyage, which took Darwin to the Galapagos. So this is the third voyage around Australia. And you get expression of GFP, and we were quite pleased with that. But you can do much better if you modify this, the 5' UTR. You can make a fiendishly efficient messenger RNA and get really bright fluorescence. And there's a difference of about 15-fold. And it was these levels which myself and Frank Sainsbury, who at the time was a PhD student with me, realised actually could be of practical importance, being able to produce quite those levels of protein. So he and another PhD student, Ava Tuneman, who actually took that photograph of the leaf with the vaccine dripping out of it, set to and made this series of vectors, very much a sort of reducing to practice in sort of patent IP technology terms. So you can make this system very usable, made these, they stand for P, easy and quick, or PEEP. Actually, after we named it, I discovered something on PubMed, which it was actually previously used in the 1950s for a measure of psychopathic behaviour. So I hope that isn't an omen. And you can make these plasmids, again, include a suppressor of silencing to boost levels, and various bells and whistles. And you just have to infiltrate those into leaves. And this is, I don't know, have you actually carried out any agroinfiltration experiments, anyone? Okay, well, you probably will. And but I'll show you what was actually involved. Now, some of this is sort of cloning 101. So, but I hope that you'll find this interesting. I made this for the public. It's quite cosmic. It was made by the same people who did the animations for Brian Cox's Wonders of the Universe. And you can see a certain similarity. So basically, you want to express a sequence. So that's a restriction enzyme cutting. That's the sound they make. And you take a plasmid, do the same thing, and then put your foreign bit of DNA, which is shown in purple in this, and it floats along. And that's a ligase. Put that in, and then you take some bacteria, and you take agrobacterium, which is soil-living bacterium. You can grow on a plate, it's fine. And these little bacteria are chemotactic and motile. And again, this is slightly fanciful rendition, as they sort of row themselves along like this. And all you have to do is take your plasmid and use electroporation, the same thing you'd use with E. coli, give it the shock, and the plasmid gets in and bounces around a bit, and then is amplified. And that's the purple bit you've inserted. And so you get lots of copies. And then all you have to do is grow the bacteria and take them up a syringe without a needle on, and then squirt them under the epidermis of a plant. And you get quite practiced at doing that. And then inside, something rather wonderful happens. There's the sun coming down, and there are the green leaves, the green cells in the mesophyll. The bacteria come, put out a type 4 secretion pathway system, and send the plasmids in. Now, they're not naked DNA in reality. So they get in, they're coated with various proteins, which helps transport them to the sun, aka the nucleus. So they get transported in here. And there they go. And they start amplifying and integrating and being transcribed and make the sun go supernova. And all the messenger RNAs come out, and in this case they're coding for viral subunits to make virus particles. And that's why they're purple, because viral subunits are always purple. And so that bit of the leaf actually goes purple. And if you were to do it with GFP or DS red or something, it would go colour. And these will actually self-assemble into virus-like particles, which you can then extract and purify. And the great thing about that, goes on, is that you can scale it. So I showed the syringe pressure infiltration there, and that's, I think that's me. Yes, which I regularly go down the greenhouse, of course. A typical day. Doing this on a single leaf. And you can reverse the process. So instead of pushing the bacteria under the epidermis, you can actually rely on the stomata to actually dunk plants in a soup of agrobacterium, reduce the pressure above, and the air will bubble out through the stomata, leave it a bit, and when you release the vacuum, it all springs back and takes up the agrobacterium suspension, so you get the whole plant at the time. And that's great if you want to do larger scale. And the companies who do this on a big scale, and this photo, quite old now, is taken from a facility in Quebec City, from Medicargo. They have all these benthomianas on a conveyor belt, and they do great batches of this by agroinfiltration. And they're particularly concerned with using this to make influenza vaccines. And there's the kind of yields you can get. So what kind of things have we been doing with this kind of system? It's called Cowpea Mosaic virus hypertrans, because it can get high-level translation. And all sorts of kind of things you can make. So you can make enzymes. This is a thing called human gastric lipase, which is used for digestive studies. You show the enzyme's active when you extract it from plants. You don't have to extract the enzymes. You can just use it for bioconversions within the leaves. And this is some work with Anne Osborne's group showing a compound which makes the leaves go purple under UV light. You can look at the enzymes of chlorophyll degradation, and let's put those in and see what they do to the chlorophyll content. And you can actually look at manipulation of secondary metabolites, and this is an intermediate of a thing called adenosine, a triterpene. So you can do all sorts of manipulations within the leaves. Now my main interest, as you probably gathered when I talked about virus-like particles and all these things, are these shells of viruses. And a virus-like particle differs from the virus in that it doesn't have the genome in it. It's non-infectious. It basically just contains the protein shell, possibly some host nucleic acid as well, but it's a non-infectious mimic, basically. And so we've used this system to make all sorts of ones. Bovine papillomavirus, I know Leslie knows that with Andy Love, did that. We've worked on turnip crinkle virus, another plant virus used in nanotechnology. Nervous necrosis virus. Audrey, you know all about that now. This is a fish virus for really great importance to aquaculture. It causes disease of sea bass, the ones we looked at in particular. Hepatitis B cause, Hadrian worked on those. They're used as a display platform for novel vaccines. A thing called blue tongue virus, much more complex, 4 different proteins in concentric spheres. Ava Tuneman, who developed the vector system, has done a lot of work on blue tongue virus. You can produce particles which will protect sheep against disease using these plant-produced. It's a veterinary vaccine. You can make empty shells, I'll come on to in a minute, of Cowpea mosaic virus. And also shells of poliovirus. And the only one I'm going to talk about in any detail, you might look at that picture with absolute horror and say, this is going to last forever. But I'm just going to talk mainly about polio, some very recent work, and a little bit just to close the loop. Sounds like that film Looper, actually, if anyone's seen that, with Cowpea mosaic virus. So, production of poliovirus VLPs. Well, I'm sure everyone's heard of polio. And everyone, I'm sure, in this room has been vaccinated with it. Probably by the oral polio vaccine, or Sabin vaccine, which is a live attenuated vaccine. You sort of drink something. There's also Prior to that, for us more senior citizens, the inactivated poliovirus, otherwise known as the Salk virus, which people like me got in the 50s, which is a killed vaccine. So fantastically effective vaccines. So why are we bothering with plant-based production? Well, there's a problem with both those. They're highly effective, but you can never eradicate polio using them. And there's a need for a new generation of vaccines. The World Health Organization has called for that. And the problem is, I mentioned that virus-like particles don't have the genome. And that's why they're much safer. They can't revert or cause a disease. But also, in this particular case, the protein-only shells aren't stable in the absence of the genome, which is if you just express the protein, what you get is something which is an irrelevant antigenic form, either sometimes known as the C or the H form, heated, is the, and it still looks like a particle, but it doesn't give you protective antibodies. And so the idea is, could you actually make a next generation of stabilized particles using expression technologies such as that which I've just described in plants? So it's a little bit complicated. To make a poliovirus capsid, you need two components. You need the structural proteins, which come as a precursor, and you can put that in one plasmid, and you need a protease to process those to get full assembly. And you put that in another plasmid. Now, the great thing about agroinfiltration is very efficient. So you can simply mix the two types of plasmid and do a co-infiltration. You don't have to put both components on the same plasmid, so you can do some mixing and matching. And you do your infiltration. This is just shown on a small scale. And we're very fortunate we have a collaboration with the National Institute of Biological Standards and Control in South Mims, NIBSC for short. And they can actually work on live poliovirus, which at the John Innes you'll probably be relieved to know we can't. So what they can do, they did an absolutely classic bit of virology. They simply passaged live poliovirus at increasing temperature. And what you do is, viruses are such a small genome, and the fact that they're made of RNA, which when it's multiplied, there's no proofreading mechanism, you acquire mutants very quickly. And this selects for the ones which are more stable, because they survive at higher temperature. So it's forced evolution in the test tube, or on the plate, if you prefer. And then that would have been known to people like Sabin and Salk, or that technique, all the way back. But now the twist is, the more modern thing is you just take your mixture of these things and you do next generation sequencing on them, and then you find out all the versions which have stabilizing mutations and identify them all. And this is the nice bit. They can just send us a file of the sequence. We never see the polio. We just get the sequence, and we just have it made synthetically by a company. And there's no problem doing that, because it's not an infectious entity, it's just the coat protein. Put it into the plasmid, and lo and behold, you get assembled particles. This is the cryo reconstruction done at Oxford. They look exactly like authentic poliovirus, but they're completely hollow. There's no genome in them. And you can actually do a series of immunological experiments, and with animal testing, you can protect mice against polio infection. So these actually do work as an efficient polio potential vaccine without any genome. Now, the other thing you might be saying, well, what happens if you just use this with wild type? Do you need the stabilization? If you do it just with the wild type virus, you don't get anything as pretty as this. You get the wrong form, and it doesn't give any protection. This is actually as good as the inactivated polio vaccine. So there's a lot of interest in taking this further and scaling it up for deployment. Which brings me to the next point, is the issue of how you go about this. And some of you may have come across this concept of the Valley of Death. Now, it is, of course, from a poem by Tennyson about the charge of the Light Brigade. But this idea that you do work in a research facility, such as the John Innes Centre, and it looks really good. You make these on a small scale, these polio potential candidate vaccines, and as you do that and you're successful, the Research Council, the WHO says, well, you've done it, thank you very much. No, you can't have another grant to just keep on doing it. You need to commercialise it. But of course, you then talk to the commercial people. They say, oh, it's a bit of an early stage. How can you prove it will scale up? and it'll work on a large scale. You know, what are the problems you might encounter on scaling up? You say, well, we can't really tell until we try some. And yet, if you're in an academic lab, you don't really have that kind of facility for sort of quality assurance and process development. So, a few years ago, and this was actually after, as Celia mentioned, the Innovator of the Year award in 2012, awarded to both myself and Frank Sainsbury, a PhD student. So you can get these prizes at an early stage. You don't have to become geriatric before you get awarded them. We decided that, or I decided that what we really needed to make the most of this and strike while the iron's hot is some kind of translational facility to work out how you could take something from the lab scale to a more commercial scale. And that's what the final building looks like when it was completed. And it was open, that's the business end. You can see it's not an academic lab, it's far too tidy for that. And the business end is growing the plants under LED in containment facilities, growth rooms, and a vacuum infiltrator to do that vacuum method on a large scale. Next to it, they have actually smaller ones now. And it was opened on the 23rd of January. Now, this is an interesting question. Who recognises that person? Fun? Joe Johnson. Yeah, who is the younger brother of Boris Johnson. You'll notice the hair and the eyes. But actually, he's not quite like his elder brother in that he's quite pro-European and he's very modest. So there's a slight, I don't know what happened to that. Makes you lose your faith in genetics, really. And me suited and booted. Director Dale Sanders and Melanie Wellen, the CEO of VBSRC, the Research Council. And of course, fortunately, Frank Sainsbury happened to be visiting from Australia. He now works in Australia. And so we were able to celebrate in true fashion. And like all people who've won a cup, you inevitably have to kiss it. So that's now up and running to produce various things. And one of their products, and you can actually buy this, well, this little bit of an advert for leaf expression system. They want to make up to 10 grams of protein and it's scalable. One of their things is to try and build on this work on those CPMV particles and to produce other VLPs. And at this point, I shall hand out some of those other VLPs we've made, all different sizes and shapes, including Gemini virus particles, some made by us, some made by Leaf Systems. And one of the things is to try and take this Cowpea Mosaic virus technology from over 20 years ago, made using infectious virus particles, to take it forward. Because it had more or less stayed there, given the size and the stability of the particles, and the size of the inserts you can make. So it's a great particle. Lots and lots of things have been done with it. Not just that vaccination, a lot of anti-cancer work, particularly from a former student of mine, Nicole Steinmetz, is stimulating all sorts of anti-cancer immune responses. But it would be much nicer, rather than using infectious virus, to actually use some kind of VLP of that. And Cowpea mosaic virus is actually related to polio, so you can make the particles in a similar way that you could to polio, although I have to say that was done before polio. Now, the problem with using the infectious particles, as we developed in the 90s, there's limits to the change that you can make. They contain viral nucleic acid. The presence of the viral RNA inside the particles means you can't put anything else inside. You can't use them as nano containers, can't be loaded, so create empty virus-like particles. And what you do there, rather similar to the polio case, you take the precursor of the coat proteins, take the protease, put them in, and you can make nice virus-like particles. Now, a critical thing to show quite early on was doing this, how similar are they to the wild type? Because if you're going to be able to use all the information you garnered with those wild type particles and apply them to the empty ones, they have to be effectively identical. You know, you could end up with a situation, for instance, like you find with polio. In the absence of the genome, there are changes. So we did have a look with our colleagues in Leeds using CRO-EM. And you may remember I showed the stuff for the wild-type particle. There's a slight difference here. You can see these magenta regions up here. They're bits you can't see in the wild-type virus. They're extensions at the carboxy terminus of the small coat protein. And as you zoom in, you get a nice high resolution structure, but there's no gold in them, they're ales. There's no genome in there. In fact, there doesn't seem to be any RNA at all. There seems to be just this virus only packages its own RNA. And that's always been a mystery. And that occurs also with things like polio. They only package their own RNAs. And it's been a mystery because there don't seem to be any specific signals, nothing you can really see clearly on the genome. So this just shows, it's basically showing off the resolution of the map. But one of the fascinating things, so we know this looks, apart from the little bit extra, it does look absolutely identical to the wild type. So you can actually use it as a non-infectious substitute. But seeing that extra bit gave a clue as to how the virus actually might assemble and how it puts its RNA in. And it's something which is really a bit of basic virology again. So having gone through all this business of using translational research, getting particles using technology for use in bio and nanotechnology, you now begin to use the information you get to actually understand some basic virology again. So you're again completing a circle. And this is this bit with the magenta bit. What it turns out, and this actually explained a lot of old data, is actually these things hold hands to form pentamers. And they're critical in the early stages of assembly. And without them, if you remove these little bits, only 24 amino acids, these pentamers don't form, and you abrogate assembly. And so you do need those. And I rather liked a Russian colleague of mine, Yulia Mesheriakova, who made this analogy, saying it's a bit like Henri Matisse's dance, a penton dance. All the subunits have to hold hands to form this penton. And that's just a bit of basic virology we've uncovered. And it also explains how you form an RNA binding surface, because the RNA, when you see it in particles, lies at what's known as the two-fold axis. And by creating these pentons first, you can create the axes where the RNA binds. So the biotech side has gone back to the basic virology side. And the interesting thing is, what you find is that the only RNA during an infection which gets packaged are the replicating RNAs. So if you do a full infection, both RNA1 and RNA2 replicating, both will go in. If you just use RNA1 in your infiltration, only RNA1 gets inserted. So it seems to be that only replicating RNAs can get put in, and you can come up with, if you make a non-replicating version of RNA1, that doesn't get in. The details are not so important here. You make lots of particles, but no RNA if you don't have replication. And so just to summarize that, if you just have a full infection, both RNAs get in, the protease and the precursor of the coat protein, nothing gets in. Have RNA1 with the precursor, and the RNA1 encodes the protease. You just get RNA1 in. If you have a non-replicating version of RNA1, has all the other enzymatic functions, has the protease, again, just empty. And various other versions of it down here, which are not important at the moment. Okay, so we now know replication is absolutely critical for encapsulation. That's a bit of basic virology. Actually, you can go around that wheel again and say, well, can we actually exploit that of a way of putting specific bits of RNA of our choice into the particles? Because nothing else will go in the plants. You need replication. That coupling, can you go back and use that? And the answer is yes. Again, this is Hadrian's work now, with both multiplying and both replicatable and non-replicatable RNA versions of RNA2. So that can replicate, that's reduced replication, that can't. And then put it in with RNA1. I won't go through all the details here. You need replication of the RNA2 to put in this artificial RNA, which contains GFP. And okay, everything works with GFP, as I said earlier. But you can actually specifically package a GFP messenger RNA, but of course you don't have to stick with that. You can put any RNA of choice in, specifically into the particles. And that can be extremely useful, for instance, if you want to make some diagnostic reagents If you want to protect the RNA you're trying to use as a positive control, you wrap it up in CPMV, coat protein, and just add it to crude samples. And that'll act as an in-tube positive control. It's an interesting thing you can do. You can also use it, say, to deliver RNA to specific cell types. And so you can imagine doing something like this with, you put your designer RNA in, here's a CPMV capsid. You could put a targeting peptide on it, so it would only be directed to, say, cancerous cells expressing certain markers. And that, we know that Cowpea mosaic virus is taken up, and you could then deliver a specific RNA. It could be an antisense RNA, some kind of therapeutic molecule. And so we've gone all the way through from basic studies, through applications, to more basic studies, to more applications. So I call that a virtuous cycle. So I was looking this up last night to make sure I got the quote right. I think it was Louis Pasteur who said, "There is no such thing as applied science, just science which is applied." So if it's good enough for Louis, it's good enough for me. So I just want to close by acknowledging a lot of people over the years who've worked on this, and there'll be people who aren't in the photograph, or I don't mention specifically, but I thought I'd concentrate on those work I've talked about. We have in the audience Hadrien Perret, who's lurking at the back there somewhere, who did a lot of work in the RNA packaging, as did Inga Cruz, now in Glasgow. Frank Sainsbury and Eva Tuneman, who developed the PEAK system, and they weren't in the lab at the time. Julia, I mentioned, with the Penton dance. Pooja Saxena, who works actually for a company using plant-based vaccines, did a PhD with me. Joanna Marzian in the front, who did all the work on poliovirus, or the JIC end, and a lot of work by Keith Saunders on making things like turnip crinkle virus. I don't think there's anyone I've missed out here. There were others as well. I have to also thank hugely the cryo-electron microscopists in Leeds for all those nice images and being able to actually look at accurate 3D structures for us. And also the poliovirus consortium from the WHO. And they're actually critical, coordinated from Leeds. Incredibly important that we have the help of NIBSC, because they can work on live poliovirus and carry out the immunological testing for us, as well as isolating the mutants. And University of Oxford for the cryo-EM here. And of course, you don't get anywhere without funding. So most of the work I've talked about is either funded by the BBSRC or the World Health Organization. And so with that, I'll finish and happy to ask questions, but I think you get a chance later to do that. But so I think you're probably desperate for a coffee. I know I am. Thank you very much.